Here we go, just waiting for people to drop in. That is looking good. Well, welcome everyone to Neuroccino on this April the 8th, 9:30. Oh my god. Very briefly, rules of the game. Anyone is welcome to present a piece of science they got excited about, and that could be a paper, a book chapter, a YouTube clip. We had a range of diverse sources of exciting science. But today we have Marcella, who is a postdoc within the team in Bordeaux, and she got excited about a paper dealing with the neurodynamics of attention. Marcella, if you want to share the link in the chat.
Yes, I will. It's not open, but I have a link with, I have a link with an open version. Let me check if I can share this with you. Let me know if it works. It's
system.
This one. Let me know if it works and if you have access to the paper. I think this is an open version. Cool. Perfect.
The floor is yours.
Cool. So I will share my screen. Here. Okay, can you, I will just try to expand. Right.
That looks very good on my screen, Marcella.
That looks good? Okay, cool, perfect. Okay, so hello everyone. So I will share with you today this paper that I found in the Nature Human Behavior Journal entitled Neural Evidence for Attentional Capture by Salient Distractors. So this paper, I attract my attention actually because in the introduction, they immediately highlight the importance of orienting towards salient objects for evolution, but also it makes the projection to the nowadays by telling that usually we get distracted when we are like doing a work or we're trying to focus on a task. We get distracted by, for instance, our phone buzzer. So the objective of this paper was to explore the neural dynamics and the brain regions in the human's brains that are responsible for the attentional capture and the ways in which these salient distractors interact with our current goals. So to this aim, they describe in the introduction a little bit the research that have been done before in this topic, but usually the main constraint is that using electroencephalography, they cannot follow the neural activity for both, so the target, the current target of the task, but also the distractors. They either focus on the goal or on the distractor, but not the interaction. So they took advantage of a group of patients, so in particular 18 patients that had implantation. So they were implanted with these intracranial electrodes for control for epilepsy, like to measure the activity of the epilepsy, the seizure. And they were asked to perform this attention task that I will show you here in the figure. So in A, you can see some trials of the task where they perform in total 6 blocks of 720 trials. And we have these two conditions. So for all the tasks, you have, they were presented with these 8 different shapes that could be either a diamond or a circle. And inside they had lines that could be in parallel, like in vertical or horizontal. And so one of the conditions was, and they were instructed to find the target and to identify if inside the target there was a vertical or horizontal line. So the target was the only shape that was different to the others. So in this case, if you see the trial, the blue one and the orange one, we can see that only one shape was a circle. So they were instructed to find this one and to say if the line was vertical or horizontal. But one of the conditions, so 2/3 of the trials had a distractor that was this one of the shape that had a red frame. So here in the same panel A, we can see immediately some results for the reaction times, where they actually validate at the behavioral level that when the distractor were present, the reaction times were longer compared to the trials where there were no distractor at all. So when the participants only focus on the target. And then in B, we can see a little bit the context of the experiments where you have the participants performing the task. We can see the different electrodes that were recording for, I guess, all the participants, the 18 participants. And during while they were recording, right, the activations of the different electrodes. And the authors calculated this time frequency. And in particular, in this study, they focus on high frequencies, specifically between 60 and 100, as this has been considered to be a logical marker of local neural populations activity, and also because these frequencies have been correlated with attention in previous studies. So they explored this high frequency, and in C, we can see actually that in both trials were distractor present and distractor absent, that there is this activity in the high frequencies. And here when you see the time, so the x-axis, you can see two lines, gray lines, and these are the significant difference in time between the two conditions. But I'm going to detail this later. And then in D, they kind of validate that the activity they observed in the high frequencies was correlated with behavior, because they show this significant correlation trial by trial between the time, so the delay in the response, and the high frequency activity recorded in the electrodes. So then we can see the method they used to analyze this task. So remember that the aim was to identify the neural activity related to either the target or the distractor. So they use this model that I will not be able to explain in detail, but you can go to the paper, of course, to know more about this. That is the inverted encoding model. So what it does, it identifies or it relates the neural activations to the specific locations of the stimuli. For instance, here, as we can see in A, we have the location 1 to 8. And with this model, the authors were able to identify the neural activity that was related to each of these different locations. So this is important because they after could identify the neural activations that were specifically to the target or to the distractor. So the locations where they presented either the target or the distractor. And they calculated, with this model, they were able to calculate this score that is called channel tuning functions that I will be presenting along through all the paper. And this slope represents the amplitude of these neural activations for this specific location. So the higher the amplitude of the slope, it means the higher activation or the neural response to the specific location. So for instance, here in B, we can see the comparison between the trials where the distractor was present or the distractor was absent in blue and orange. And we can observe that there was a higher amplitude, so higher activations, we can say, of when the distractor was absent compared when the distractor was present. at this very early period of time between 200 and 300 milliseconds. And and here in the right side, we can see this latency. It means that it represents the moment where the slope reaches the amplitude. So if the latency is shorter, it means that the slope for this condition was reached an amplitude before compared to the second condition. So here we only are saying that the amplitude of the slope when the distractor was absent was reached before compared to the condition where the distractor was present. And this is only following or tracking the target activity. So it only means that when the distractor was absent, the attention to the target occurs before. And then in C, we can see this tracking to the target and the distractor simultaneously. And what we observe here is that in the electrodes where they analyze the activity in C in the left side, we can see that the response to the distractor occurs before, while at the same time, the response to the target was decreased. But then over the time, we can see that after around 600 milliseconds, the response to the target increased and the response to the distractor decreased. So what they show or demonstrate with these results is that when you have a distractor and a target, you first pay attention to the distractor, and then over time, you can recover this attention to focus on the target. And in the right side, they only demonstrate that because they only took into account the electrodes where they could register high frequency activity. So here they only demonstrate that for the electrodes that they didn't consider, there were no difference between the distractor or the target. And then in D, they compare the responses that were fast and slow in time in the conditions where the target and the distractor were presented simultaneously. And what they observe is that in the fast responses, the attention or the neural activity related to the location of the target was higher along the time. However, in the slow responses, we can see that the first attention was directed to the target, to the distractor, and then to the target. That's why, or that's related, not causal. That's pattern relates with the slower response times of the participants. And here in E, what they show is that there was a negative correlation between the reaction times and the peak of this slope and a positive in the target. So it means that the shorter reaction times had a higher peak in the neural activations related to the target. So participants focus immediately to the target, so the response times was faster. And then when they compare this peak of activation related to the distractor, they observe a positive correlation with behavior. What means that when this the neural activity or was higher in the location of the distractor, participants were slower to respond to the target. So this is an overview of all the analysis they perform, trying to follow the dynamics for the target and the distractor. And then they try to associate these neural activation to specific brain locations. And so then they describe this network that is called distractor-associated brain networks that are different brain regions that were identified as contributing to these different neural activations. So they use an atlas to identify different regions and brain networks. and they identify brain networks that were related to the DMN, so the formal network, and limbic that were related to this contribution. So then I will not talk about this part of the figure, but this is only a replication of their results using some other different analysis. And here what we can see is the results at the brain region. So they explore 8 different regions that were identified as contributing to this interaction between the distractor and the target. And so in this analysis, what we observe here, we observe the slope in green, and they run again the different analyses, and they observe how this attention to the target it's affected or impacted by removing the activity of specific regions. So what we see here in A, the description of this analysis. So if the distribution moves to the left, it means that the region that they remove has a major contribution to processing the distractor. And if it moves to the right, it means that the region has a minor contribution to processing the distractor during the task. So here they evaluated these 8 different regions that are the amygdala, superior temporal gyrus, middle temporal gyrus, anterior cingulate cortex, hippocampus, inferior parietal, precuneus, and prefrontal cortex. And what they observed, this first row of regions, so the one in the colors of red, they highly contributed to processing the distractor during the task, and in particular, the amygdala and the superior temporal gyrus. And then they observed that these other 4 regions that are below had only a minor contribution to processing the distractor. And then here in the panel C and D, we can observe the replication of these results using different analyses, where they show the higher contribution of these specific regions and the lower contribution of these other 4 regions. And what they also observe is here in D, that they replicated using the activity of these different regions. They replicate the results that they showed before without relating to the brain. So, right, the results I showed previously. So they replicated that there is activation in time in this period between 200 and 400 milliseconds. And here we can see in red that these regions with the major contribution has a higher activation, so a higher slope, compared to the regions that has a minor contribution to processing the distractor. And then, so again, most of the analyses are for replicating on the different results that they have been showing. And so here, this is the last figure. And what we have here in A is that, again, if they take separately the activity or the neural activations for these 4 different regions that are major contributors to the processing of the distractor, they observe, again, this higher activation between the 200 and 300 milliseconds. And on the right, what we can see in the latency is that the peak of this activation, so of this slope, is not different between them. So what they conclude is that these 4 different regions actually are acting in parallel, so as a network, and there is no specific delay on time when they contribute to processing these distractors. And finally, here what we can see in panel B, they explore the temporal similarity across all the different regions that I already mentioned in this period of time where they observe the difference when processing the distractors. And what they observe is that the anterior cingulate cortex had higher similarity with all other regions, and in particular with temporal brain regions. And so they highlight this region as a hub to processing the distractors when performing this attentional task that is in interaction with the temporal regions. And then they, in addition, explore this temporal similarity between the prefrontal cortex and all these major contribution regions, and between the inferior parietal and all the major contribution regions, as this, because the prefrontal cortex and the inferior parietal has been highly related to processing targets during attentional tasks. And what they found is that the prefrontal cortex has this a higher temporal similarity with these regions, the temporal regions, and with the anterior cingulate cortex. So what they conclude is that there is this network that is where the anterior cingulate cortex is a hub that is related to the temporal brain regions to processing the distractors, and that the prefrontal cortex make the link between the regions that are minor contributors to processing the stimuli and the regions that has a major contribution to processing this kind of stimuli. So as conclusion or discussion of this study, they first they highlight the importance of exploring the high frequency activity that is linked with behavior that we observe in the task of attention, and that they capture when the salient distractor is included in a task. They observe that the neural activity changed in around 220 milliseconds, where they see that the attention to the distractor increased while the attention to the target decreased. And of course, they highlight the importance of these 4 brain regions and the role of the anterior cingulate cortex as a hub. And so, and the last main point is that there is a huge debate on the literature about if the attention to the stimuli, either a target or distractor, is something that occurs automatically, or if it occurs more in a more controlled way. And these results are supporting the idea that they occur more automatically. And yeah, with this result, they say that we should revise all the theories that relates to the attention and brain. And I think that's all that I can tell you about this paper. So happy to ask and receive any questions if you have.
Thank you. That was great. Hands are coming up. I'll hand over to Chris. Oh, hello. Sorry, Chris.
Thank you for presenting that. I don't know if you know, I think it's Fristen's model of how the brain is modeling the world. So basically, I think the idea is that the brain is constructing a model of the world, like how we. So it's it's predicting what's gonna happen, and that when you have something that is not in what you predicted, that's how you react. That's gonna create this reaction and then trigger like responses or whatever. Do you know if with this kind of experiment we can tell if it's just a reaction in the like typical way that we see? Is like, oh, we see something that is, well, in that case, yeah, I what I mean is I don't know how to explain the reaction to a distractor in another way that it's something that was not predicted by the brain. It was unexpected in the experiment. But I don't know if they talk about this this idea. Like, is it do we react to the distractor because it was unexpected, or is it just a salience effect. It's like, oh, we see it and we react to it, but not in term. It's not a prediction that the brain is doing.
Yeah, thanks for this question. I'm not, well, they don't discuss about this in the paper. What I think they control for, I guess it's like with the all the EEG tasks, also with the even response potential, evoke potential, where they control for the balance between the different conditions. So what with this, they try to control that the possibility of having a distractor or not is the same. So I don't know, and I don't think, but not sure, if it relates to this like this difference between what the brain predicted and what was presented. What they discussed, it seems to be more than it's just only because there was a salient stimuli. Because in the discussion, what they talk is that for these different theories supporting the idea that this is a very automatic process, also because it occurred very early in time, and we are like measuring the activity in the intra-EEG. It's what they support is that it can be either the attention for a target, for a distractor, for anything that is a salient stimuli. So I would say it's not something that is a prediction, but I'm not sure.
So it could be an evidence against the model of prediction.
They say it's not a controlled process. I don't know if it's the same. I don't think so. But I'm not. I don't have the knowledge to say that it's against that.
Well, they don't talk about it, so yeah.
Exactly, and they don't talk about that. So I'm not sure. But it's a good question. We can discuss more after.
It's a very long debate. Thanks, Chris. I think Eduardo had his hand up first.
Yeah, I have a question. It's probably more directed to Chris, to Marcela, about this stuff about the prediction model. Because I guess at a certain point, the participants are doing an experiment where they are expecting something unexpected. So how does the model it by Preston deals with those kind of things? Right, because they're not really, it's not really an unexpected stimuli coming out. It's kind of expected, even though you do not know exactly what.
Yeah, as they don't talk about this in the paper, I wouldn't be very exhaustive talking about. I don't feel very comfortable saying that it's against or is considering these kind of predictions. What I can say is that yeah, the two conditions were the same, and the balance between the trials, and as with the model that they use, they are able to identify the neural activations for each of the different locations. So what I guess is that, yeah, I can only argue about what they found, that is that these activations to the distractor occurs before, where the ones to the target are decreased at the same period of time. But I don't know if they are related to something that the brain predicted and occur or doesn't occur, and then they don't discuss about it. So I don't know exactly if some of the experiments they did, some of the analysis actually are trying to control for this. They don't say anything about this the theories of the prediction. So I cannot say if the results actually are against or in favor of that. I don't know if you have any.
Sure, thank you. It was just a curiosity that popped up in my mind after the.
Yeah, I'm
That's
sorry I
great.
cannot help with that, but yeah, it's interesting.
And Muge has a question for you as well.
Yeah. Hi, Marcella. Thank you for presenting this. So I guess I'm a bit puzzled about the spatial selectivity of these channels. So these different electrodes. So they present the distractor and target in different locations, right? And then does each channel have a preference for a specific location, like this? Like, what is this spatial selectivity that there?
Yeah, this is something, and maybe also for Eduardo and Chris, that is described in the method. Maybe in the method they use, they don't mention that, of course, explicitly, but maybe it can be useful to understand if this model actually controlled for this prediction. But what it's explained in the model that is not very easy to understand for me, at least, because it's very like mathematical formulas. But what each channel represents are the activations of a group of neurons in the brain. But they don't describe if there is specific channels that are associated to specific locations. They only, they use a function to select the different channels. But what I understood from the model is that that each of the channels represented the neural activity of this population for a specific location. But it's for me it was difficult to go through this model. So I just took the main points that were the activations related to the location that were specific to one location, and that the slope represent this amplitude of activations for the different related to the different locations. But
they don't fixate the head, right?
No.
In the visual fields, they can look at different locations.
Yes, no, maybe this is the important point. I'm sorry, they were instructed to look at the cross-fixation. So that's why I guess they can do this relationship to the different locations, and also that's why they selected this method, so these kind of trials, so they could identify these different locations for all the trials. And always the participants were looking at the middle of the trial, so at the cross. But yeah, so here in A, you can see the model, and you can see here the different channels. So I guess they use the sum of the different channels to identify the neural activations for each location. But yeah.
Yeah, to me, like, it was strange to see like STG has some kind of location preference in the visual field. So that's okay. But yeah, I'll take a look at the model.
Yeah, they don't look they don't do the relationship, at least in the main paper, the main manuscript, they don't do a relationship between specific locations and brain regions. Because also the patients, they had to do a random group analysis, because the patients, they were 18 patients, and they don't have the electrodes in the same place. So had to make some average and some different group of the electrodes in. So yeah. I guess that's why you cannot relate brain and location, because it changed across participants.
Okay, thank you.
That's all the questions we have time for, I'm afraid. But there's a vivid debate going on in the chat about
I'm not follow.
flexibility. I'll send it to you later, don't worry. We may actually turn this into a nightcap discussion because it's quite interesting what's going on here. Thank you so much for presenting the paper. I wish you all an exciting week in science and see you next Monday. Bye.